Hello students, welcome to SK Biology. Today we are starting second important point from this 7th unit, which is molecular basis of inheritance. We already studied classical genetics, where we came to know that genes are fundamental units of heredity. But actual nature of genetic material is not known. To study molecular basis of inheritance, first requirement is discovery of genetic material. Number of scientists worked on it. Here in this image you can see that scientists quest for genetic material. They all contributed in the discovery of genetic material. In this table I have summarized scientists and their contribution in the discovery of genetic material. We will study all these scientists' work in detail one by one. Let us begin with first scientist, Friedrich Miescher. In 1869, first time he isolated DNA from pus cell. While working with pus cell, he found that these pus cells have some molecule containing high amount of phosphorus. He named it as a nuclein. Further, scientist Altman found that nature of this nuclein is acidic. So, he named it as a nucleic acid. Today also we call genetic material as a nucleic acid. After the work of Miescher and Altman, for lot of years there is no any development in the discovery of genetic material until scientist Friedrich Griffith gave the direct evidence of genetic material by transformation experiment. Griffith worked on pneumococcus bacteria. He found that in the bacteria two phenotypes are present. One is with smooth surface S3 strain, they are virulent in nature, and second is with rough surface R2 strain, they are avirulent in nature. In his experiment, he injected virulent S3 bacteria into healthy mice, and he observed that all mice died due to pneumonia infection. In second step, when he injected a virulent R2 strain into healthy mice, there is no death of any mice. Further, he heat-killed S3 virulent bacteria to destroy their virulent nature, and then these heat-killed cells he injected into healthy mice. He observed that all mice are live, and these results suggest that heating of S3 bacteria reduces virulent nature of these bacteria. Now he combined a virulent R2 and heat-killed S3 for injection into mice. As in individual experiment they both are unable to kill the mice, here also it might not kill any mice. But surprisingly, Griffith found that when mixture of R2 and S3 heat-killed bacteria injected into mice, some of the mice died due to pneumonia. These results conclude that there are some component present in S3 heat-killed bacteria that transforms a virulent R2 into virulent S3 bacteria, and that molecule must be a genetic material.